A very welcome to all of you. First, let me ask, am I audible properly?
Yes, you are.
Okay, thank you. So, very welcome to all of you, and good evening, good morning, or even good afternoon, based on wherever in the planet you are in. So, joining live here today, we have with us Dr. Supriyo Das, Assistant Professor, Presidency University, and it's my great pleasure to introduce him in front of his in front of this audience, as when I was about to start my journey about in geologist, when I was in the very earlier days, not I would say as career, but in my student life, in my BSc days, I got himself as faculty member. So I was very lucky to have you, sir, today. So, as of like me, he was also a student of Presidency College, the then Presidency College, now Presidency University, Kolkata, from where he completed his BSc and moved to IIT Roorkee to have his MTech degree, and then went to Stockholm University to gain a PhD degree. And after that, for postdoctoral fellowship, he got several awards and postdoctoral positions, and also some prestigious awards like British Council fellowship, then Swedish National Academy fellowship, and also in EAOG scholarship. And then, after completing his several postdoctoral tenure, and he came back to India and joined as an assistant professor at Presidency University, Kolkata, and where he is still now he is working on that. And he mostly focuses on elemental, molecular, and stabilized geochemistry to understand the interaction between the continual changing climate, environment, and human activities. It's a very interesting thing, and we will definitely hear from him. And also, the major part of his research focus, that he was a member of IUDP Expedition 354 and the 38th Indian Scientific Expedition to Antarctica. And I know that you are waiting for him to hear about his Antarctic journey. And also, so, sir, the stage is up to you.
Thank you, Suham. Uh, well, you gave a very long list of my struggle. Uh, I really thank you and this opportunity to present my talk. I can see some of my students are here as well. So let me share my presentation first. Yes. So I hope you all can see the slide. Is it correct? Can you see the slide?
Yes, sir.
All right. So as Suham introduced myself, I am an organic geochemist, and I have been to Antarctica in 2018-19. So I'll talk about my journey and a little bit about the scientific discovery that I made over there. And this is all about uh, sunscreen lotion. You know what? Whenever we go to beach or a place where we can have some UV radiation, we can have the possibility of exposure to UV radiation, we apply sunscreen lotion. There are various print of sunscreen lotions. And after reaching Antarctica, I was very specifically told that never go out under the sun without having the sunscreen lotion. I even had one friend in South Africa who had this skin cancer. So we applied sunscreen lotion whenever we went for field work. And later, I realized not everybody has that uh, option to fancy a sunscreen lotion whenever go out. And that happens with the biota living in Antarctica. So what would they do? So that was the question was in my mind, and I actually worked on it. I got the opportunity to work on it a little bit, and I will tell you what actually I found. So as I said, I am an organic biogeochemist. Basically, I have geology background. That means I apply certain tools in organic biogeochemistry, mostly lipid biomarkers and stable isotopes, to reconstruct past climate and past environmental changes. Now, in Antarctica, I went to study the lake sediments and soil sediments and the organic matter preserved in those sediments to find out whether the organic matter is keeping a record of the climate change, the ongoing climate change we are talking about every single day. The Earth is getting warm, and we are adding carbon dioxide to the atmosphere, and that is the cause of the global warming, etc. So I wanted to see a record of these changes in Antarctica. But after I arrived in Antarctica, I was a member of 2000 of 38 Indian expedition to Antarctica, and I was there in 2018-19. It was summer. It was very sunny and very warm. Trust me, it was really warm. And after I arrived in Antarctica, I found a very interesting feature of Antarctic biota. Now, in Antarctica, you won't have large land plants. You won't have any plants as such. There is no tree, there is no green, and that is very interesting, and that is very uh, particular for Antarctic training. I mean, the people who go to Antarctica, they were specifically told that you won't see any green for months, and that affects the brain. So we knew that there is no green at all, but we didn't know there are plants, lower plants, moss and algal mat. So I saw them, and they were very beautiful to some extent. So I was uh, staying at a research station in Antarctica, which was called Bharati. So Bharati Research Station is in Larsemann Hills, and Larsemann Hills is in the eastern part of Antarctica. Now, if you know, there are two research stations for India. One in Larsemann Hills, that is called Bharati, as I said, where I stayed for about 3 months, and there is another one which is a little bit far from Bharati, quite far from Bharati, and that is called Maitri. So first, whenever we go to Antarctica, we stay at Maitri for a few days, and from Maitri, we go to Bharati. So whoever working in Bharati stays there, whoever working in Maitri stays in Maitri. So I worked in Bharati, so I was stationed at Bharati. The Bharati research station is very uh, modern. As it looks, it was started having guests from 2012, and it has all modern facilities, everything that you want, high speed from high speed internet to modern toilets and everything. Now you may find it very interesting to when I say it has modern toilet. Well, that's another story for Antarctic exhibitioners. Now, I was at the eastern part of Antarctica, East Antarctica, which is called Larsemann Hills. The Larsemann Hills has many islands, and it has about 150 or even more small lakes and ponds. So my work area was around the ponds and lakes and within the lake water. So there are two peninsulas, as you see in the picture on the right, Groundness and Brockness. And there is a large island called Fish Island, and another one in the north, say Ilzakit Island. So there are many other islands which doesn't have a proper name. And this area is ice free. I mean, that sounds very peculiar, right? Antarctic expedition, and I'm talking about ice free zone. That is correct. In summer, in austral summer, that is when it is winter here in India, it is summer in Antarctica. We have complete ice free area in Larsemann Hills. There is no ice cap. The snow that accumulates during winter or rest of the time of the year melted down, completely melts down during early spring and summer, which lasted for a few weeks, probably not a month and a half, but everything goes away. So what we can see is that they are in raw, and that is red. So no green, only reddish terrain and green ocean, sorry, blue ocean and blue sky. Now, there are several ponds and lakes. The, the ponds are say shallow water bodies, I should say, uh, that is formed from the accumulating meltwater in shallow depressions. So there is a pond in the picture on left, and there is a shallow lake in the picture on the right. So lake is called mud water. I don't know why it is called mud water because the water is very clear, and there are several ponds, uh, as you can see here. So there is no rainfall in Antarctica. That is very interesting. What we can get is only snowfall. The snow accumulates at certain places, and when it gets warmer, the snow melts down. The snow meltwater accumulates in shallow depressions, forming ponds or even forms shallow lakes. And during the winter, the lakes were frozen. The top of the lakes is frozen. If it is not a deep lake, then the entire water mass can get frozen if the water remains that long. And in summer, coastal summer, as I said, the temperature really goes up. It was about 2 degrees Celsius, and I could not tell to my friends that it is 2 degrees and it is so hot. You can literally come out from the station and walk around in a t-shirt. But we are not allowed to do that. That was different thing because the Antarctic, we call harsh climate in Antarctic. That doesn't mean that it is always harsh. The temperature can fall very fast. And if there's a blizzard, you can simply die. The temperature may look like 2 degrees Celsius, but you will feel it minus 20. So accident happens. We had to take precaution every time we go. And this time, I'm not talking about temperature. I'm talking about UV radiation and sunscreen, right? So let me talk about lakes a little bit before we come into the finding. We call these lakes oligotrophic. That means you won't see any phytoplankton production in the surface of the water. The water is very clean. You can actually see the bottom of of a lake, which is say 30 meter deep, and that is scary. I can tell you. If you see that deep, the bottom of the lake, you feel scared. At least that is what I felt at some point. So the water is very clear. There is no phytoplankton, but some lakes have benthic mat. We don't say it algal mat because the composition is not always algae. Those are benthos, the mat that we can see. And the moss, we can we have abundant moss beds along the meltwater channels. So, I mean, through the channel, the meltwater comes to the lake and the shallow depression forming the ponds. Along those channels, we can have the moss. So these are all we have when we say plant species in mainland Antarctica. There are birds, as you can see. There is a skua sitting in the picture on the left picture, but those are very rare. Apart from penguin and skua, there are no Antarctic life. Of course, I'm not considering the human visitors. So just after arriving in Antarctica, I saw the moss beds, and they are very colorful. Fewer red, fewer reddish green, and fewer green. Now, with time, I noticed that the green moss beds are turning into reddish green and eventually red and then gray. And finally, before I left, they become almost black and very dry. So I started wondering why it is so. Now, moss, if you know, if there is any biologist here, he or she will know more about moss than I do because I'm not a moss specialist. But what we know about moss was that they are non-vascular plants. Like the plants you see all around, those are vascular plants. The moss is non-vascular, lower plant, we call them. So they grow when the soil or the substrate is moist, and when it becomes dry, they actually become hard, dry, like they started hibernating. We call them desiccation states or moribund states. Whenever they get water again, they rehydrate and become gray. So that is their normal cycle, at least in Antarctica, and it is well known. Now, there are certain scientists who work with mosses in Antarctica. They say that this change in color actually for two reasons. First, they want to protect themselves from water stress, that is like low availability of water, and the other is they want to protect them from UV radiation. Now, there are certain biomolecules which actually uh, are synthesized by moss for protecting against UVB radiation. Those are called pectin and phenols, and there are some pigments, carotenoid pigment we say. Those are for changing the colors. Here you can see the green moss, which is growing in a shadow. So this moss has become reddish green and finally become moribund when I catch the return flight from Antarctica. So this is what a moribund moss look like. You see, these are like blanket on the floor, right? These are quite hard. If you take a slice, you can this if you consider it as a cake, you can take a slice and you will see the leaves and different structure of the moss. Now, this stage of moss indicates there is no water left. So they are sleeping here for the next winter rain and the spring to arrive when they will get water and they will be fine. Now, this change in color, this change in form, morphology, we know why it is happening, and we know little bit about the molecular level differences in green moss and in moribund moss. But we don't know actually the function of those molecules. Say, if I go under sun, I will become tan, right? So my skin will become darker. See, I'm an animal, so it is a different thing. But if I think it in another logical way, that the change in my skin color is a change in color of the pigment, right? So here, from green to red, that is change in color, pigment color, we call it carotenoid pigment. That means they are producing more carotenoid pigment than chlorophyll. So that part is well understood. Now the pectin, phenols, all those other molecules, they are also being produced when when the morphology is being changed. But where is the connection between water stress and UV radiation exposure and these molecules? That part was missing. So anyway, there are lot of lichens, as you can see. So that makes things very complicated because when we say moss or benthic mat or lichens, those are not like a single plant. These are like consortium. There are a lot of cyanobacteria, algae, diatoms, lot of things are living in a consortium. So you cannot simply say that the moss is changing color or lichen is changing color or mat is changing color. There are individual organisms in there. So they all have different way to respond to a external stressor. So everybody is responding against UV radiation or water stress, and they have their own way to survive. So we see a mixed signal at the surface, a change in color, for example, or change in morphology. But every single organism within that consortium is involved in that change. That makes things quite complicated. So anyway, uh, what we know about Antarctic ozone depletion is that it is an old story. We all know that stratospheric ozone is being depleted in Antarctica, and there are some sort of anthropogenic and natural reasons for that. I will not go into this. Uh, and whatever I told you about moss, that is also very much well known. You will find if you simply Google, you will find a lot of article on that. What additional I see is the benthic mat. Now I wanted to work in the lake. So when I started working in the lake, I found the lake bottom have very thick benthic mat community. And those benthic mats, they are also having different colors. The deepest one are green, and the benthic mats who are at the shallower depth are reddish green, even gray. Now benthic mats, they don't have any issue with the water. They are always underwater. So they don't have any problem having water stress. What they could only worried about is UV radiation. The deeper benthic mat may have little UV radiation. The shallower parts, the benthic mats, they are getting higher amount of UVB radiation. So that is the only thing I could think of at this moment. But they are also showing the same change in color as you can see in the moss beds. So I started collecting those samples and I did a lot of reading over there and I brought back those samples. So far, what we what I understood that both the moss and algal mat or benthic mat, in other way of putting it, they produce UVB protective molecules. We call it UVB screening compounds. They produce that, and they are depending on the species and the morphology and the consortium uh, thing. They produce different amount of these screening compounds, but we don't know what are those compounds. What is the composition, molecular composition of those compounds? And I wanted to study alkanes. The reason I wanted to study alkanes is that if you go out and if you see any plant, you will have alkane in it. You take a plant leaf, you see the waxy coating on the leaf, it has alkane in it. We use alkane for paleoclimate reconstructions. We use alkane for uh, paleo-environmental reconstructions, and we study alkane for all sort of organic geochemical analysis. So that is very common molecule that we study, and it is very logical for me to start with alkanes. Now, some of you who doesn't have a chemistry background may worry what is an alkane. Now, alkanes are hydrocarbons. Those are made up of carbon and hydrogen. And they have a single bond. They're bonded with a single hand to each other, and that is why they are called saturated. So if we are having a saturated hydrocarbon like this here, we call it normal alkane or n-alkane. What we study here in Antarctic samples are the distribution of long chain versus short chain in alkanes. Now, what is chain length? Here you see the N C 21 to 30 at the bottom of this figure, right? So C stands for carbon, and 21 to 30 indicates the number of carbon in that molecule. So if an enalkane has carbon molecule ranging from 17 to 20, we call it short chain enalkanes. If it is 21 to 30, then it is long chain enalkanes. So what we did, we actually studied the different ratio of long chain versus short chain. And the reason we did that, because every single plant has its own signature of enalkanes. The large plants, woody plants, for example, it's just an example I'm giving you. They have more long chain enalkanes. The lower plants like algae, moss, they should have shorter chain enalkanes. So that is a very standard understanding, but it is not always true. But in any case, every plant has these two sort of range of enalkanes, and we can always have a ratio of those enalkanes. So in moss, lichen, and algae that I collected from Antarctica, they have enalkanes from C17 to C30, and we use different ratios. Some ratios are called odd over even predominance ratio. So I'll not go into that. And some called the carbon preference index ratio. So those are different ratios. Those are used to study uh, the relative distribution of in alkanes. So I will not go into that today because that is not actually the part of the story that I am going to tell you here. Now, alkane, they are known to play an important role against desiccation. The large plants, the terrestrial higher plants, I should say, they produce inalkanes as a mechanism to survive in low water conditions. Even in moss and algae in different part of the world, they produce inalkanes for both for, to survive in low water conditions and to protect themselves from UV radiation. But their mechanism is slightly different from Antarctic moss and algae. So this is a very schematic diagram. Here you could see the short chain inalkanes and long chain inalkanes ratio in the vertical axis, and in the horizontal axis we see the green mat and red and gray mat, and their concentration and the concentration of inalkanes in those algal mats. The green mat, you see that bar is quite tall, that indicates very low amount of short chain inalkanes, and the gray mat has the highest amount of shorter chain inalkanes. So with decreasing water column depth, we could see higher amount of shorter chain inalkanes in the benthic mat. So as I said, there is no issue with water stress. The only problem they are facing is the UVB exposure. So they want to protect themselves from UVB exposure, and what they do, they started producing higher amount of shorter chain inalkanes to create a sunscreen lotion for protection against UVB. For moss, it was not that simple because the green moss, they have water, and if they are under shadow zone, then they don't have much UV exposure, right? But it is not always true. They do have UV exposure. Is the rock that you can see in the picture was not good enough to protect them. Hello, to protect them against UVB throughout the day, and it is sunny, 24 hours sun. So you cannot have protection from UVB. What you could have, what the green moss had, water for some time. When they have water, they were green. When the water started diminishing, they started changing their color. Now, as long as they have water, they probably have some mechanism to protect against UVB. When they have less water, they then they were in trouble. Then they started producing more and more shorter chain inalkanes to protect against UV radiation. But at some point, they didn't have enough water to do that as well. So they are started dying, and they don't want to die because without water, nobody will survive. So they want to take care of the water stress. They wanted to survive in no water condition, and they started producing more and more long chain inalkanes to go into the modible stage. So you can think it in that way. As long as there was enough water, they didn't take care of much UV stress issue. When the water become lesser and lesser, the UV stress become more prominent. When the water become very little or almost dry, then they started producing preparing themselves for the dry condition, and UV stress become insignificant to most. So the way the inalkanes are produced is it's just like they produce alkyl molecules, and from alkyl molecules inalkanes are produced. I'll probably not discuss much about it. There is a way that mosses produce inalkanes because it is not straightforward as we can see for benthic mats, because they have water stress to handle and UV stress to handle. And depending on which stress is more prominent at that particular moment, they change the synthesis of n-alkanes. Whether they are going to produce long chain or short chain depends on the primary stress condition. So in brief, in this figure, we could see that benthic mats are primarily dealing with UVB stress, and moss are primarily dealing with the UVB and water stress, both. So in conclusion, I could simply say that n-alkanes are produced by both moss and benthic mat, and depending on the primary stressor, they produce either long chain n-alkanes or short chain n-alkanes. But both can be produced at the same time. In-alkane synthesis is primarily controlled by UVB radiation in benthic mat. In moss, it is both controlled by UVB radiation and water stress. And when they become moribund, they don't care about UV stress that much. They just want to protect themselves from water stress. They don't want to dry and disintegrate. So they just want their molecule to remain intact and stay alive until the next spring when the water for melting snow packs comes to save their life. Now, when I started this talk, I actually said that I am an organic biogeochemist and I wanted to reconstruct past climate, past environment. And here I am telling you, or I told you, the story is about moss and benthic mat. So I'm a geologist. I didn't go to Antarctica for studying moss and algae, did I? No. So where is the connection? Why a geologist? Why actually forget about the geologist? Why we should care about uh, UVB radiation exposure to moss and benthic mat in Antarctica? What's the significance of this study? Now, the significance is several-fold. The ozone depletion is not an issue only for Antarctica. We are having it right in tropics, where a large population, human population, survives. Right? So, if we are going to have more and more UVB exposure, how our body will react? We'll have more sunscreen. Well, that is one part of it, but algae and moss are not animal like us. So we cannot say that enalkane will be produced in our body and we will get protection from UVB. So that is not going to happen. But it may affect our food production, our agriculture. The plants, if the plants are going to produce a different amount of enalkanes, so I don't know how it will affect the crop yield. It can affect us. Now, from a geologist or paleo reconstruction point of view, we use enalkanes for past environmental climate reconstructions, and we say or we consider that certain chain length of enalkanes is produced by certain group of plants. Now, if that plants were exposed to UVB at that particular time, geologic time, so millions of years ago, we don't know what happened at that time. So if the plants were exposed to UVB and they categorically produce a different amount of short chain or long chain in alkenes, we have that significantly in the in the geological record. But we are not considering it. So that would be a very interesting fact if we can reconstruct past events when we have ozone depletion, and we could use that for prediction what will happen in future. If the UV radiation, if the ozone become more depleted, we are exposed to more UV radiation. The plants are exposed to more UV radiation. Then how they will react, how they will protect themselves, what changes they will happen in their body, and how it will affect us in terms of food production. There's another interesting part to it. You know, ozone depletion has to some extent connected to volcanic activities. So past volcanic activities may cause ozone depletion. Even the volcanic activities these days, uh, they can also affect the ozone layer, stratospheric ozone layer. So if we are constructing past climate associated to past volcanism, we should look into ozone depletion. And n-alkane study would be a very good way of doing that. There's another very interesting thing. We know when we study archaeology, when we study human uh, evolution, we used to be hunters gatherers, and then we started doing farming. We become civilized. If we are civilized today, so it didn't happen in one day. We don't change our habit of living just because we just want the change. There's some reason. Now, there are various ideas, there are various opinions why human population increased, and why we moved from hunter-gatherers to farming activities and so on. So I will not talk about that, but there are certain time when human left Africa, and the cause for that we don't know. There are certain time when we had a population bottleneck. We don't know the cause of it. Some of us probably know the Toba volcanism, the Indonesian volcano Toba, and we may have a thin layer of glass of volcanic ash in our sediment. We use it as a age marker, 75 kilo years, right? So Toba volcanism is often linked to human population bottleneck, but there is no direct connection. Why a volcanic activity reduce the population of human uh, species? We don't know whether it has something to do with the ozone depletion and UVB radiation. So those are the things uh, we may explore, we may think for future study. Here I will finally stop. Uh, this is the paper we published on our finding. So these are all published, and these are the students who work with me. I mean, I just went to Antarctica. I brought the samples, but then the students took over and they worked. They are colleagues of mine, junior colleagues, who helped me, and that's a work. We call it a consortium again. So we worked together. We found something very interesting, and here I told you the story. You may like, you may not, but it's important that you understand. And please ask me if I missed anything, or if I didn't explain something clearly. Thank you for listening.
Thank you, sir, for this wonderful talk. Yeah. Thank you, sir, for this wonderful talk. So, first, I would like to say something to the participants that one feedback form has been given in the in-call message section, and you can just fill it up with your comments and feedbacks about this EPS3, how we can improve it, and what topic do you want to hear in future, and all that. And also, people can ask questions in the message section, and I will try to put them through to Dr. Das here. So, just let us begin with our discussion session. It's our custom to have a detailed discussion session after each talk in EPS3. So, first, sir, a very blunt question. Like you may say it a student to teacher question, as all know here, I said it earlier that you were my teacher during my BSc. So I learned some new techniques and tools, and we were seeing that you were the first person in the department when we were there as student, that who did something that was not what we call the core geology subjects, not structural geology, not petrology, not sedimentology, not paleontology. You do organic geochemistry. Okay, so biogeochemistry. But definitely, as a geologist, when you are selected in this 38th Antarctic expedition, you went there as a geologist. Fine. But was it in your mind that you thought of this project that I would work on moss? And if that, so why on earth a geologist like you, who studied geology for 5 years and worked in PhD and postdoc in biochemical processes in sediments, now why would he or she look for mosses in Antarctica where the rocks are abandoned?
Well, Subham, I should confess I didn't go to study moss and algae in Antarctica. As I said, I wanted to reconstruct past climate, environment, everything from lake sediments. But after I went there, I did the study. This is still ongoing. I found the mosses, the changing color of mosses, more interesting. I mean, I found it more interesting. And as Bharati station had high-speed internet, I started studying what is known, why they're changing the color. And I found a missing gap. I mean, missing information, a gap in research. And apart from doing the lake sediment coring and soil coring, all other sampling, I started studying on moss. So I didn't know anything about moss or benthic mat before going to Antarctica. I mean, I have seen moss and algal mat, but not the way or not exactly the same species, I should say, as I seen in Antarctica. So I didn't know anything before going there. I started studying on them, and this way of sampling moss or any bionic material is different than sampling sediment. So I am an expert in sediment sampling and rock sampling. So those are not a big deal for me. So for moss and algae, you cannot simply collect and put it in a bag and take back to India. So it was different. So I had to study, I had to ask around and develop way to get those samples intact back at home. Because if they degrade, the study will not of any use. So all these things I had to develop. So it was not in my mind. And sometimes I tell these days I tell to students that you may be given a project, but you may end up doing something more interesting. I mean, everybody does paleoclimate study, and I'm yet to develop or yet to get sufficient data from Antarctic sediments where I can use it for real paleoclimate or paleoecological studies. But what I have today, nobody has found it. So even a biologist who is studying Antarctic moss and benthic mass for decades, they didn't think it that way. So I don't know whether the alkanes, the distribution alkanes I found uh, will be supported by further study or something like that. Or, but that is a real finding, right? So that is more interesting even today. So I think it is a puzzle uh, that I tried to solve, and I'm to some extent successful in that. And nobody has thought about it before, so it is very accomplishing or very satisfying to some extent. So I did something new, very challenging to me at least, and like uh, yeah. So I think that was a take-home message to students here I want to share that just keep your eyes open. Probably something more interesting you will find, and you'll get involved into. And you may need to study, you may need to develop yourself. I mean, as I was not a biologist, I didn't know how moss looks like. Everything I learned by myself, and you can learn it anyway, and you can find something new or something more interesting. I don't know, Subham, if I answered you
No, yes,
correctly.
sir. That is true. That to learn is to be human. So we learn every day, and we study, and again we learn, and then we evolve. So you touched upon human evolution and out of Africa theory that 75 years ago, 75,000 years ago, sorry, a bunch of people from Africa, they started moving out. So we don't know what driven them. Like something like that. Though they were about to start some new venture there, but it failed. And like you, they must have started mosques outside Africa. But yeah, let that crap happen. But a technical question. So you said you were about to collect the sediment samples there, and you just come up with the idea about moss and algal mats. Just be stationed at Bharti station. Okay. So how can you manage to collect the sample? I know I faced the similar problem while working on my research topics. Sometimes that we are about to collect some mineral samples. Okay, but somehow I just wanted to see the algal mats there. So I work in vicinity of hot springs. So there are typical algal mat structure presented there in many hot springs what I encountered. But there we had no experience first, and second the expertise, and three the technicalities to collect the samples. So it was the fellow biologist in the team who supplied you with the sample. What could I say? That sample collection techniques and tools and their preservation. The main thing is the preservation. Am I correct? Because it's not rocks that you just break it and collect it with your sample bag, Ziploc bag. It has to be preserved, intact, and it has to be alive uh, up to you collect them and take them to the lab to be analyzed. So what was that?
Sampling in Antarctica is always very challenging. Like even if you are going for rock sampling, you have to trek far away. You have to camp in sky under the sky. So it is always challenging, and the lake sampling has different challenges. I'll come to moss later because when I mean here you don't have a boat. I mean, a boat may not be always useful in certain lakes, and the water is so cold. I mean, I had the experience of putting my hand under water while collecting the benthic mass as well, and you have this burning feeling. It's so cold. I mean, 4 degree temperature is not very cold if you have it in Kolkata, but in Antarctica, when the wind speed is very high, you feel it like burning effect. So it was very challenging. So going into water and getting the sample from the water under the water is always challenging. So we had some inflatable boats, and then the helicopters. They actually dropped the boat on the lake surface. Uh, so we had that's a different, I think, part of a talk how we actually sampled the algal mat from the lake bottoms and underwater sampling all these things. So once we have those samples, we had to preserve them in certain solvents. So those solvents were there. I mean, we have a very modern lab in Bharati. So it was not a very complicated for sample preservation. We preserve these samples in -20 refrigerated freezer, I mean, and then there are ways to transport the samples by ship, or even I could have my samples with me with sufficient protection. So that is how we transported the samples. It was quite challenging, but the protocol is always there. The most challenging part to me was sample collection because in one case we had the inflatable boat which was about 70-80 kilogram, not very heavy but heavy enough, and it was huge, and it had some metal structure in it, so you cannot carry it on soldier. So the helicopter need to be there. Helicopter comes with a ship, and that comes at the end of the expedition, like towards the end of the expedition during March or in February or something like that. It came a little earlier, so we had the opportunity to have the helicopter. Helicopter dropped that inflatable platform. We better say, not the boat, inflatable platform on the lake. But the lake started uh, freezing by that time. It has a very thick uh, layer of ice. So we ended up on a ice on a lake. So it was not water, and it was so transparent. That is what I say, that I could see the bottom, 30, 40-meter deep lake bottom. I could see, and it was so scary because when we on that all means like three of us on that platform, and we dragged the platform in the middle of the lake. That ice, the thin ice, this thing started cracking. So you feel like you are on a glass, plate, in the middle of the glass plate, and the glass is cracking, and you are about to fall. You had this feeling. But anyway, we had to break that ice and get our sampler down. Hello?
Yes, sir, I am here.
Yeah, so we had to get our sampler through that ice. So at some point, we felt very scary. I mean, we felt very scary that, what happened if we all fall in the water, and like we'll die in few minutes, something like that. So those are the challenging part.
So I didn't see any questions till now in the messages. So students and participants, don't hesitate. Just raise a question there if you have any. So coming to my next question, a technical question. So you talked about UVB. So what does this B signifies? So I'm asking as a geologist point of view, who don't have the knowledge of this kind of thing. So
I think, Shubham, you know more about A, B, and C kind of UV than I do. So
so
UVB is very harmful. It damages the DNA. That is something I learned. So UVC, I think, is used for killing bacteria, virus, something. So I am not an expert in UV radiation. Uh, so I can only tell the wavelength or sort of things, but I am not
here
much
main okay, okay. Let us
knowledgeable.
just go apart. Another interesting fact you showed a bit in the concluding slide. You said that these changes in planet, the planetary atmosphere and their paleoclimate can be traced back via archaeology. Okay, because and it's your expertise and your interest, research interest also engulfs the interaction between climate and human habitability. So how and I know that you worked in some archaeological projects also. So was those projects was only simply the dating the artifacts, what we do in using the stable isotopes, or from that you are about to trace back the archaeology, and how trace back the paleoclimate, reconstruct the paleoclimate.
See, the human climate or human environment interaction is very interesting. So my involvement with archaeologists is not about studying their material, although they want me to study their artifacts. But my interaction is mostly to understand why, on first place, humans started living at some place, right? So there are many school of thoughts. In India, for example, we always think, when I was a kid, when I was at school, I used to learn that human civilization started along the river, like Nile or any other rivers, right? Like uh, how to call this? I sometimes forget these names. But anyway, these are river-borne civilization, but which is not always correct. As for Indian subcontinent, uh, it is not always correct. We have villages, not cities. In big like big built cities, we have large villages habitats along lakes or large ponds, and that we found several places, and that actually drew my attention, my interest, and then I started working with archaeologists in coastal West Bengal, and there I found that people started building huts like structures someplace, and probably they abandoned that place due to some storm or cyclone activities. So those things are those records are there in the sediment. So we wanted to know why. First thing, why people started living on that place, and why all of a sudden they left that place. So those are the things connected to climate and environment. And when today we have, I mean, if we have a cyclone here, we always like, okay, it is devastating, it damages houses and kill people. So did it happen back in in history? And if it happened, how they affected our livelihood? That is something I study, and my interest is only in Indian archaeology because there's a lot of scope to work. Nobody has worked that much. Outside Harappa, there is little work. It's open ground.
Listening to your answer, I was smiling because you said that you studied the paleoclimatic conditions within the lake sediments and all. So, as a researcher of planetary science, mostly who works on Mars, so I invite you to explore the Martian lakes and their lake sediments. So there are quite a good amount of data in terms of the rover data. So you could see the different layers and the lake deposit, the lacustrine sediments there, and even you could get to see their characteristics and the mineralogical and elemental compositions to some extent. Yes, of course, you don't have the samples right now here on your hand, but they are good data. So please explore that. We need good scientists and geologists like you to be explored in planetary science because apart from Earth, there are much possibilities in to work rather and to get habitable zone for us in future in different planets. So I would yes I would like to ask our director to post a question here. You'd but let me ask one more question before that. Like, what's on your take? I started talking about planetary science, but what on your take about this study of astrobiology and planetary analog in Antarctica? You see, there is a huge gulf in recent years that Antarctica must be treated as an analog to typically for Mars, as it was untouched from the past since the Gondwana breakup. It got separated in the southern zone and covered with ice. So the rock record and there the lacustrine sediments, the seasonal variation there, they produce a huge amount of what we call sort of Martian minerals also. And these extreme conditions, okay, and their uh, flora and fauna, and particularly the this extremophile life, they are sort of interested the scientist, and also it may be a proxy to the ocean worlds and the icy worlds like Europa, Ganymede, Io. So what's your take on that?
I think, like as you said, it is not a new idea to study Antarctic sediments or the primitive life forms in Antarctica, and making it as yeah, I don't remember the term you said, like analog. I think you said of
Yes.
Martian life or
Yeah.
basically life in Mars or other planets. So that idea is not new, and I know people are working on this line, not from India, I guess. But here, I think we should study. We should start studying because our the stations that we have in Larseman Hills. I mean, if I take a picture and say this is Mars, you'll probably believe that this is Mars because it is red rock all around and no life. I mean, no visible form of life. If you don't see moss or mat, so that is the place I think we should study. And the planetary scientists, they should uh, initiate some sort of experiments over there. Like if they're planning to drop a rover in Mars or even to Moon or wherever, so they can try it in Antarctica, in Larseman Hills at least, because it's a hard rock. So how the rover will work in that hard rock may have some sort of analogy to the rover, to the same rover working in Mars or any other planet. So I think that's a very good laboratory for experiment. Other than that, even if we don't have samples, but we have data from these expeditions, those can be analyzed and studied. But I'm sure that a lot of people are doing that. So I'm very curious to read their finding and see how things are uh, working in planetary science. I just want to know more about it.
You correctly said that people are working on that, and have declared Antarctica as a test bed for the future planetary missions. But yes, there is a lacuna in our country. So planetary scientist and not we cannot bind someone like planetary scientist. So geologists, biologists, physicists, everybody should come up and come up with some new ideas and take the direction to the future. So now I would request our director, Dr. Priyabrata Das. He raised a hand, I see, to pose his question. Priyabrata Das, please unmute and yourself. Yeah.
Thank you, Subham. Actually, it is not my actual question. Actually, Antarctica is a fantastic place. Other than geological potential and this environmental, what's the climate change and all these issues, it's a test bed for like the kind of we are using nowadays, and also for to you can use that this moss and algae people are nowadays using. There's that potential to use this thing on in international space station or in moon if you want to set a colony or want to set some laboratory there, whether we can use these mosses because they are highly affected by the radiation, uv radiation, and in space also we have high radiation effect. So whether so is there any studies going on in that direction also that we have to think from Indian point of view, because India also thinking for this lunar expedition this year, I think Chandrayaan 3. Maybe in future also we are going to send some astronaut in international
And
space
don't
station.
forget the Gaganyaan mission, where
Gaganyaan
this
mission.
kind of
So this kind
expertise.
of expertise, it means to test the genetical thing of this mosses and all this the algae, and you have to select those particular that can generate huge amount of oxygen for sustainability, space sustainability. That because I am asking this question because I know there are multiple students from astrobiology group are here in our seminar, and they are eagerly waiting for this type of questions. And I think if they are interested to work on this particular domain of research, I think they can work. I think, and this is a separate thing. It's biology, and then genetics, and also geologists. They have to combine together and their application in space. So that kind of thing also we can think.
I know some of my colleagues, they are studying the bacteria, some sort of bacteria
Yeah.
uh, living in those moss and benthic mats, and that can be used for purpose like for producing medicine or something like that. So I don't know much about their research, but we are looking for many things in those moss and mats. And I think I'm quite sure somebody is thinking that line that if we are uh, starting our any settlement in other planets or outside Earth, probably those are the plant species from which we could uh, develop something, some sort of large plants or something for food production or something like that. Because normal plants, they will not survive in extreme
mm,
conditions, and
mm,
these are the species they are surviving extreme. I mean, the extreme climate doesn't make sense if you just see the pictures. Once you go there, then you will. When it may be very warm and sunny and very pleasant, you want to have a barbecue outside, but
mm,
in next moment you will have these blizzards, and then 9, 10 months there is no sun at all, and when there is sun, it is all over the day. So if you are getting something, you are getting at the extreme level. So life is there at that extreme level.
extremely.
So that is very interesting, and I
Mm,
think like uh, we can learn from those plants, and we can take it from those plants if we are having some settlement, in another planet. I think somebody, I'm pretty sure somebody is thinking that sometime. Even if it is not from Indian
yeah,
scientists, but somebody is doing that.
that's why I can remember the last in Chang'e mission, Chinese mission last time. They sent some cotton seeds to moon, and they sent the photograph of that sprouts. Okay, so they are also trying to uh, start this kind of experiment in unknown place in out of Earth environment. So it's a good
Yeah.
opportunity actually. If you think towards that direction also, it will be good.
Uh, Thank you, Dr. Priyodas.
Mm,
You participated and this interesting discussion session. I wish it never ends, but we have some time bindings. And I see one question from one of our participants. It's Dipali Singh. She asked that this talk was about different mosses being resistant to UV radiation, right? So you said in your talk that the exposure to UV was a stress situation for them rather than water. Was their genetic component studied by you or your team or anybody to confirm this, like activation of stress gene?
Yes, we did environmental DNA study just to identify uh, uh, the structure of this moss bed, as there are lot of organisms living in the consortium. The interesting thing is that we didn't see any DNA damage. I mean, the genes were not damaged uh, by UV radiation. So definitely they were protected, and there is nothing like UV resistant moss as far as I understand. Uh, all mosses are protecting themselves from UV radiation, and how they are doing it, that was the question. So probably they are uh, producing more short chain alkanes, or they are doing something else to protect their genetic structure from UVB damage. So that is all they are doing. So because if we say that one species are resistant to UV and another is not, that species is gone. So it is not like happening yesterday. It is happening for last few decades, right? So if something is not if it all UV resistant, it would have extinct by now. And there are certain, you are right, there are certain species who are more UV resistant and certain who are less. When their relative abundance changes, the green species, for example, that will diminish when the water stress will become more and more prominent, right? Because they cannot protect themselves against UVB radiation. So that is correct, but it is not correct that some are totally resistant and some are not. All of them are more or less resistant.
Thank you, sir. And I think Dipali's questions has been answered quite elaborately. And thank you again for your time and such wonderful talk. We'd like to hear from you in future again in
Thank you,
a
Shubham.
different topic. This time, probably on your IODP expedition, that Indian Ocean Drilling Project. So, thank you, and thank you all the participants for your time. Good night.
Thank you, Shubham. Good night.